So, hi everyone. My name is Ian Murphy, and I'm going to talk a bit about my life, uh, how I got up to this point in my grad program, but also, my journey through science and a bit of my research as well. So, I will start with saying that I was born in Massachusetts, in some small town no one has heard of, probably, and I went to high school in that same town no one has heard of, and I decided to travel pretty far for college, so I went to UMass Amherst. Partly because my sister had gone there, so it felt a little familiar. Um, and I went in as a physics major, and that's what I graduated in. But before that, I did leave the state and I went to Scotland. I studied abroad at the University of Glasgow, so I saw a lot of cool things. I saw the architecture, I watched a rugby match, I got appendicitis, and I was adjacent to some pretty neat things that other people were doing, which was great. So I'd recommend studying abroad if you can. But I did come back and I graduated from UMass Amherst with a bachelor's degree in physics. And I wasn't really trying to go into school again yet, so I got a job and I worked as a printed circuit board designer at a company in Wakefield, Mass. Called Newgrange Design. And I worked there from 2019 to 2021. So through the pandemic, luckily everything we did was virtual anyways, so it was a pretty seamless transition to home. But I had a lot of fun there. I just knew that I wanted to go back to school eventually. So I did end up applying for grad schools in physics, kind of biophysics as well, because I knew that was an interest of mine. And I ended up at Brandeis and getting my PhD in physics, biophysics, kind of. I'm in my fifth year, and hopefully I can graduate next year, but we'll see. So as I mentioned, I ended up kind of in biophysics, but I actually started in like microbiology in science. And when I think back about why, I'm like, oh, it's because both of my parents did that. So it's something that was pretty familiar to me. So I was like, okay, I'll explore that. So I kind of started with microbiology and I kind of bounced around to like virology, immunology, kind of biochemistry. And then sort of around middle school, late middle school, early high school, um, I was watching TV and I was like, oh, I want to be a surgeon actually. And to start, I kind of was like, maybe I'll do oncology or something. Kind of just like medicine though. I was watching a lot of medical shows and I was like, oh, okay, yeah, I'll do that. Um, and I bounced around. I did end up landing on surgery, as I said. And this was going into college. So right when I graduated high school, I was thinking about surgery. And then I realized surgery is not for me. It's it's actually probably really hard. So I actually ended up watching a documentary on particle physics, and I was like, I want to do that now. So I went into college med as a pre-med, and then I decided to switch to physics right before I got there. So I ended up wanting to do nuclear physics, and this was like kind of closer to what I actually wanted to do. Um, and then I was in a nuclear physics lab for about 2.5 years, and I realized that that is actually not for me. Um, just doing the work made me realize this. The concepts are really cool and everything. Like I want to know about it, but I don't want to be the one to do it. So I tried my hand in like a material science polymer condensed matter kind of lab as I finished college, undergrad. And I was getting a feel that this was closer to what I wanted to do, but not entirely. And then I took the 2 years off to work for a bit, and just like reset. And then I came into grad school with like, okay, now I can like really hone in on what I want to do maybe. So I ended up kind of in the biotechnology area with my research actually. And it feels more right than anything before has felt. So I think I'm on the right track. And I've actually done a bit of a loop here, which is kind of funny. So maybe I'll end up back at microbiology. Who knows? But I don't know. I might want to do this. We'll see. You know, it's all just like a journey. So I'm figuring it out still. But that's sort of how I ended up here. doing my research that I do. So I'll talk a bit about the research I do. I use very small things to make very small versions of important materials. So important materials we all kind of probably know about are protective gear, whether that's running shoes, which have soles that are good for impact absorption as you're running, because you don't want your feet to hurt, joints to get messed up. And then, protective equipment for like hockey players or football players, in the helmets, they have these pretty important materials that do a lot of impact absorption. And what is common to a lot of these is that they use auxetic materials, which means that if you expand or contract this material in one direction, it will do the same action in the opposite perpendicular direction. And this is good again for impact absorption and energy absorption. So I want to do this, but I want to do a small version of this. This is kind of like macro scale over here. Um, so I want to work on the nanoscale. And to give a sense of what that is, these running shoes, protective equipment, they're kind of around here on the nanometer range. It's kind of just like a foot long or something. And then this dot on the screen is about here on the scale. The width of a human hair is over here. Down there is the size of coronavirus. And then I want to work down here. So this is electron microscope data of a subunit made out of DNA that I'm going to explain. And the side length of this triangle is about 50 nanometers. So pretty small. Um, and how we make this thing is important to get to, which I'll get to in a second. But maybe you can already see the connection between what's going on with this microscope data and the auxetics is that I want to recreate that auxetic sort of triangular subunit material using these actual nanoscale subunits. So how do we make these things? They are made out of DNA. It's called DNA origami. And just as a reminder, DNA is what, is our genetic information. It's in all of our nuclei, and it's double-stranded, and it has base pairing. So it binds in a sequence-specific way. So G binds to C, A binds to T. And this is useful because if you have a long strand of single-stranded DNA, this black strand, for instance, and you introduce a smaller red single strand of DNA that has two regions that bind to these other two regions on the black strand, it can pinch that black strand together from disparate parts of itself. And to expand this a bit, if we just make this long black DNA a loop, we can introduce a strand that binds two different parts of it together, and it'll pinch those parts together of that loop. So the red is now the double-stranded section. And then if we just introduce a bunch of these strands, which we call staple strands, we can program them to fold into certain ways we want. And the dude who discovered this, like 20, 15 years ago, he made a bunch of cool, interesting nanoscale designs just using DNA. So all you have to do is mix DNA together under certain conditions, and it kind of does the work for you. So long as you've designed it well using a software that we can use. But I wanted to make the auxetics with this, and I was able to make sort of the open structure when it was at its most expanded pretty easily. So you can see here that the triangle subunits are bound at their vertices. They're bound just using, DNA complementarity again. It's the same concept. And, this was great. We just kind of mixed these triangles together and let them hang out for a while with each other. And eventually they did this, but we do want to close these things because we want that auxetic mechanism to occur at the nanoscale. So what we do, which you can't tell from this, is that on the edges of these triangles, there are more single strands of DNA coming out of those edges, but they do not react with anything yet. But if we introduce a linking DNA strand that can grab the single strands coming out of one edge and the single strands coming out of a different edge, an adjacent edge, it can bring those two triangles together and close this whole system. And when we do that, we do see that they do close. So we are pretty far along in this nanoscale auxetics idea. So now we just need to, try applying it to different aspects of, I don't know, material science. I'm losing my train of thought. But, um, DNA origami, these kinds of nanoscale assemblies have, lots of potential applications in material science again. Like, we don't really know what exactly yet, but that's sort of the idea behind a lot of science is that you just kind of do things for the sake of knowledge, for like understanding systems better, not necessarily for like some real world application yet. But the idea is that eventually it will have a world application. So, who knows? In a few decades, maybe this will be like the standard of antiviral care or something. I don't know. But yeah, thank you for watching.